Welcome back to our session entitled Current Innovation, AI in Pathology and Current Innovations and Future Visions. We've assembled a really extraordinary team of folks who are going to share their business models and some of the learning that they've gained in the field over the next 40 minutes or so. And I'd like to begin with four introductions of the folks who are going to be participating in this session. First, let me welcome Mike Montalto, who, pardon me, Mike, I hope I've said that correctly. Montalto is the Chief Scientific Officer at PathAI since November 2019, where he uses AI-powered pathology to support the development of new therapies and diagnostics for patients. Mike has significant experience in developing digital pathology diagnostic products and driving cutting-edge translational research in large biopharmaceutical settings. Prior to joining PathAI, Mike led the majority of clinical biomarker central laboratories in translational medicine at Bristol Myers Squibb in support of all therapeutic areas for global clinical trials, precision medicine, and companion diagnostic development. Joining Mike is his colleague Cristin, who is the Director of Clinical Science at PathAI. She has 15 years of experience in clinical research through industry and academic clinical practice. Prior to joining PathAI, Cristin worked as a consultant for Beckman Coulter in establishing the clinical organization of the Clinical Decision Support AI business unit and had previously led global clinical strategy at Smith & Nephew. Welcome also Dr. David Klimstra, who is the founder and the Chief Medical Officer of PaigeAI. Dr. Klimstra received his medical degree from Yale University and also completed an anatomic pathology residency at Yale. Following a fellowship in oncologic pathology at Memorial Sloan Kettering Cancer Center, he joined the faculty of that institution. He served as the chief of the surgical pathology service and then chairman of the pathology department, and also was a professor of pathology and laboratory medicine at Weill Cornell Medicine. He worked with Dr. Thomas Fuchs in computational pathology using machine learning to develop diagnostic and predictive algorithms from pathology digital images, which in fact led to the founding of Paige. With Dr. Klimstra is Dr. Joel Oakley, who is the medical director of biomarker development at PaigeAI since May of 2022. His role at Paige is to leverage machine learning and artificial intelligence to find new complements to existing biomarker strategies with digital image analysis as a way to accelerate personalization of oncology treatment. He is a molecular genetic pathologist who has specialized in biomarker development across multiple therapeutic areas. Welcome to everyone, and thank you so much for being here today. We have about an hour together. 45 minutes or so is really for the teams here to share their experiences, their knowledge. We're going to have a natural dialogue between the teams, and then we've left 15 minutes at the end for audience questions. Dr. Klimstra, we're going to hand the mic over to you, and you're going to give us a bit of a primer on digital pathology before we get into this session. Thank you so much for that.
Thanks, Anne-Marie, and it's great to be here, and it's great to share the podium with our colleagues from PathAI. I'm experimenting for the first time with this. Oh, good, it works. It just disappeared. Okay, so this is basically to bring everyone up to speed. I'd like to just spend a couple of minutes talking about what is digital pathology. And of course, pathology, as you all know, is based on the examination of tissues removed at biopsy or surgery, and has long been a very well-established process of embedding in wax, making glass slides, and examining them with a microscope. And this has been the state of the art for about 200 years, meaning that we really haven't advanced technologically in quite a long time. Recently, we've developed the capability of scanning these glass slides with a slide scanner, which produces a digital image. And this was finally FDA approved for use in clinical practice, but only in 2017. And so the adoption of digital pathology is in fact lagging quite a bit behind other areas of medicine. And it's interesting, if you look at the rationale for digital pathology, there's actually quite a number of great opportunities in terms of maintaining the slide custody, simplifying slide reproduction and sharing by telepathology, has potential for reductions in costs. And as we're all going to talk about, it enables us to use those digital files for AI. There's an enormous amount of information contained in a digital slide. A single digital slide has more data in it than an entire MRI scan. And in fact, all of the data on ImageNet is the equivalent of about 485 digital pathology slides. So the amount of information is extraordinarily valuable. So why haven't we been doing this? Well, there are a number of impediments. It's expensive. The scanners are expensive. You have to modify your infrastructure. Importantly, you still have to make glass slides in order to scan them, so you're not saving costs there. And there's also been a bit of a challenge in getting adoption among pathologists. The analogy of herding cats sort of comes to play in that, but we're gradually making inroads. And I think the importance here is that applying artificial intelligence allows us to create value from these digital images that can help drive adoption. So how would we apply artificial intelligence for pathology? The first use case is called diagnostic decision support, helping pathologists to find the quintessential needle in the haystack a little more rapidly and more reliably than they can do today. So supporting standard diagnoses, standardizing subjective assessments. Pathology is notoriously subjective, and there's not good user agreement. And finally, quantifying things, standardizing measurements, quantifying immunohistochemistry that you'll hear a lot more about. But we also have the opportunity to develop digital biomarkers. And these can be of two flavors. We can either replicate existing biomarkers, or we can create novel biomarkers for things that we can't detect today with current technologies. So at Paige, the philosophy is that we're offering an end-to-end solution. Paige basically accommodates any slide scanner. There are many different models on the market. We upload those images and then allow the AI, whether it's our AI or whether it's AI from any other vendor or institution, to run on those images. And then they can be viewed in our custom viewer to allow output for pathology practice or for pharmaceutical applications. I'll give you an example of one diagnostic AI that we have created, and then I'll turn it over to my colleague, Joe Oakley, to talk about some of the biomarkers. Our approach to diagnostic AI is to use massive quantities of data. We have over 5 million slides digitized available for training these models. And the prior approach, which called for pathologists to individually annotate structures on a slide, really isn't amenable to working with these large data sets. So we've been using a process called multiple instance learning, in which the computer basically learns by itself what's important on the slide based only on the overall diagnosis for that slide. Doing this, we were able to create models. This one is for prostate cancer with a very, very high predictive value. This is the area of the curve you can see is very high here, and identifies foci of cancer. And by training on thousands of images, we're able to create models that generalize what we call clinical grade AI. This work was published in Nature Medicine based on the study of 44,000 whole slides, and importantly, from many institutions around the world. So we've encountered all sorts of different artifacts and can generalize to it. And you can see in studies working with pathologists who are actually using AI on test cases, we're able to reduce the error rate by 70% in the sensitivity for diagnosis. And that is in prostate cancer, whether you're a urological pathologist or not. So this has great potential to improve patient care and patient diagnosis. In fact, this is now the only AI for pathology approved by the FDA. We had breakthrough status and got our marketing authorization in September of 2021. And in fact, when they approved this, they established the parameters to evaluate AI for pathology in general. This is data from a real world study looking at the use of this product in the wild, so to speak, by an institution in Portugal, and they demonstrated a significant reduction in the need for immunohistochemistry and second opinions in prostate cancer diagnoses, documenting the efficiency of using these kinds of tools. At Paige, we're now developing decision support diagnostic algorithms for a variety of different scenarios, including prostate with grading and perineural invasion and breast cancer and lymph node metastases. And many of these are already CE marked in Europe. So this is the work that's being done at Paige for diagnostic AI. And I'll turn it over now to Joe Oakley to talk about our H&E based biomarkers. Joe, please take it away.
Not a problem. Thank you, David. So at Paige, a lot of the biomarkers that we are currently looking at are designed to look primarily at H&E as the digitized slide that we're looking at. And of course, part of the reason is that every case is going to have a digitized H&E, or at least it's going to have an H&E slide that you can digitize to be able to apply machine learning and artificial intelligence to it. And the reason that we have focused on H&E is, as David mentioned, the image that you get, even the H&E image, is extremely information dense. And we like to think of looking at the H&E image as advanced phenotypic analysis. And so when we throw around terms like artificial intelligence, machine learning, and advanced phenotypic analysis, it sounds like we're doing something radical and new. And I would argue instead is that what we're doing is making techniques and applications that have stood the test of time, newer, better, and more relevant. So, for example, we know that image on the right is an example of invasive ductal carcinoma of the breast. And we know that because pathologists over the years have looked at hundreds and thousands of examples of this and have recognized the features of this image that tell us that this particular sample of tissue contains an invasive ductal carcinoma. And they're looking at features like changes in the nuclei, its ability to form glands. Are those glands in the correct place anymore? No, they're not. They've got a stromal reaction that indicates that this is a malignancy. And we know from clinical association that this particular pattern that we notice as pathologists on the H&E is associated with metastatic potential. So we know that this is an aggressive malignancy and we can make this diagnosis. Now, we've gone beyond this as technology has advanced, as we've added new ancillary techniques like immunohistochemistry, genomics, and the like, to be able to drill down further on what is essentially phenotypic information that we're getting, the way that these cells look and the way that they're interacting with the stroma around them. And so we're still doing this initial diagnostic phase as pathologists. We still use the H&E and our own human pattern recognition of what's going on in the phenotype of the H&E to triage these cases. But increasingly, we need more specific phenotypic information. And that's where we're turning to these ancillary techniques. So for this invasive ductal carcinoma, we know that we need to look at ER and PR and HER2, at least by IHC, because that's going to have prognostic as well as therapeutic implications for this particular patient. We might look at K67. In certain clinical scenarios, we might go to PCR-based or sequencing-based genomic analysis. And so the key to remember, though, is that we're doing all of these ancillary tests just to gain additional information, deeper information about the phenotype. And that's, of course, because the phenotype, as we mentioned, is extremely information rich. The way that these cells look on the H&E is actually the sum total of all of those things that we are testing by ancillary techniques. The way that this tumor looks and is moving through that stroma is a reflection of the tumor's genome, the mutations that it has, the way it's expressing that genome, the timing and the exact way that that is interacting, as well as any post-translational or epigenetic modifications that are going on. The way that these cells look and behave is also going to be captured on this image, and it's going to incorporate cell signaling both between the stroma as well as distant. After all, estrogen and progesterone are not typically made immediately next to this particular tumor. So you capture microenvironment information, the specific cellular components, as well as non-cellular components. And you will also be able to capture the dynamism of the immune reaction that is going on around it. So I would argue that as we're examining this phenotype with some of these newer tools, we're not attempting to replace pathologists. We're really taking the old H&E based analysis and turning computation loose on it to identify new patterns that maybe our human eyes could detect, but computer eyes can now pick up. So as David also mentioned, the way that we think about biomarkers falls into two general categories, what we call replicative assays as well as novel assays. So this, of course, is where we're using H&E in a replicative case to do faster, less expensive, and more reliable screening for existing biomarker solutions. So for example, we might try to screen cases for a particular mutation pattern, such as EGFR testing in non-small cell lung carcinoma. We'll talk about one application for that in just a moment. And then we also have novel applications, which is where there is a pain point that has no or significantly limited or existing biomarkers to inform. And so one application, time permitting, would be our HER2 complete algorithm, where we are specifically looking with an IHC0, a place where HER2 immunohistochemistry currently cannot accurately go, to try and find clinically relevant HER2 expression. So some examples of these that we've developed here at Paige are in the process of developing. So one example of a replicative digital biomarker example, again from H&E, is an algorithm we've developed to do EGFR mutation screening. And what I'm going to present to you is actually taken from a publication by spearheaded by one of our founders, Dr. Thomas Fuchs. And so what this is of course, looking at EGFR mutations in non-small cell lung carcinoma. The intention that you take the diagnostic H&E, which all of these tumors are going to get, and you run an AI algorithm on that diagnostic H&E, which is going to provide an EGFR mutation score, or a likelihood that that particular tumor contains an actionable EGFR mutation. And when you do this, you can effectively start to triage for those cases on the H&E that have EGFR mutations, which saves both in terms of turnaround time, cost, as well as tissue, which is a real concern when you're talking about relatively small biopsies for lung carcinoma. So you can basically take those cases that have a very high probability of EGFR mutation screening and now go on to definitive testing to identify the exact EGFR mutation that is present and triage the patients effectively that way. And then, of course, those that are low probability can go on to be tested for some of the other clinically relevant targets within non-small cell lung carcinoma. And so the way this was developed is basically a standard approach that we take at Paige to this to avoid some of the gritty detail. And also because I'm not one of the AI scientists, we'll be relatively brief on this. But the idea here is that we have an image that we have taken from the diagnostic H&E, and that is going to be put through what we call a neural net feature extractor. So the image is going to be broken down by the computer into smaller tiles images. And it's going to do what's called a gated multiple instance learning, which is basically weak supervision. So when we train this model, all that we told it is that there is non-small cell lung carcinoma here and what the EGFR status was. And then the model will actually teach itself to recognize non-small cell lung carcinoma, as well as any features, if they exist, that would indicate that there is an EGFR mutation that is present. And then this particular model also incorporates clinical information. It turns out the accuracy of the model was improved by also telling the model, after it has done its gated attention network step, if the patient was a smoker or non-smoker, which is what you see factored in over here. And after that, it comes up with the ultimate case level classification of whether this particular patient's tumor is likely to harbor an EGF mutation or not. And so when we did this, what we found is that we can identify a signal indicating with reasonably good area under the curve, about 0.879, those cases that harbor an EGFR mutation. And the AUC is actually pretty good across the major therapy-associated EGFR alterations that are present. Most importantly, though, we did a little bit of analysis in terms of how this might affect clinical trial enrollment as well as patient care. So in the US, about 16% of all non-small cell lung carcinomas will harbor some form of EGFR mutation. And if you do conventional screening, which would be to just go to either PCR or next generation sequencing, you would capture that 16% for sure. But if you pre-screen, you can actually increase the hit rate by triaging those cases that are likely to harbor an EGFR. And this has very important implications in terms of clinical trials. So for example, if I was running a clinical trial that wanted to target 400 patients with an EGFR mutation in Roland, if I did conventional screening, I would have to screen somewhere between 2,500 to 3,000 patients to find those 400 patients to enroll in my study. If I take the diagnostic H&E, run our algorithm ahead of time, and then only send on to definitive EGFR testing those patients that were positive by that H&E AI-assisted screen, we can actually get to our 400 patients with only 1,000 screens, which saves a tremendous amount of time and money in terms of clinical trials. And in a clinical scenario, you can do the same sort of thing. If you're in a resource constrained environment and you want to be very cost conscious and time conscious, you can use this algorithm clinically as well to identify those cases that have a high probability of EGFR, so that you can focus EGFR diagnostic testing only on those cases that have a high probability of harboring a mutation. So we also wanted to briefly mention similar kind of replicative screening assay, again on scanned H&Es, this time in colorectal carcinoma, looking at microsatellite instability high testing. And I'll try to be very brief on this, not just because brevity is the soul of wit, but because we have a legal department. And so they were a little limiting in what we could discuss on this particular application. But suffice to say that you can, as you would imagine, given the known histologic association of microsatellite instability within colorectal carcinoma, train a machine learning algorithm to identify those cases that are highly likely to harbor MSI-H, which has consequences both for Lynch syndrome on the medical genomic side, as well as therapeutic implications for selection of immunotherapy for these patients. And so even a baseline model here, which is the sort of proof of concept effort at this, can identify those cases likely to harbor MSI high with really good sensitivity and specificity, area under the curve of 0.9. And so similar to what you can do with EGFR, you can screen colorectal carcinoma cases and triage those that are highly likely to harbor microsatellite instability high, and send those along for the definitive but slower and more expensive immunohistochemistry and either PCR or sequencing-based testing to identify and confirm microsatellite instability. So lastly, I'm going to try and very quickly go through this. This is an example of a knowledgeable digital biomarker example, focusing where immunohistochemistry cannot currently go within HER2 in breast cancer. And so the main crux of the problem here is that the old way that we defined HER2 positive breast cancer was focused around a class of drugs typified by Herceptin and Perjeta. And these were drugs that block overactive and overexpressed HER2. And so the immunohistochemistry that was developed for them was skewed towards identifying those cases that had overexpressed or amplified HER2. And so this would be IHC3 plus or IHC2 plus with amplification by in situ hybridization, whatever your favorite form of in situ hybridization happens to be. And so when you apply that criteria, there's only about 15% of breast cancers that would qualify for Herceptin and Perjeta. That would be what we called HER2 positive. Well, now there's a newer class of drugs that have shown clinical efficacy and are currently labeled and available to treat a different category of HER2 positive breast cancer. These are typified by Enhertu, which is an antibody drug conjugate, where instead of relying on blocking overactive and overexpressed HER2, it uses HER2 expression to guide the antibody drug conjugate to the tumor to deliver payload. So instead of having to block overexpression through the tumor, it just finds only enough HER2 to be able to find, bind, and deliver that payload. And it's the payload that's going to be doing the damage to the tumor cell, not blocking the HER2 pathway itself. And so because of this, it is clinically efficacious at much lower levels of HER2 expression, what they've called HER2 low classification, which is IHC 1 plus or 2 plus without amplification. Now, the problem, of course, is twofold. First, that used to be a category that we called negative, and the antibodies are not exactly designed to be sensitive and easily replicative within that particular new 0 to 1 clinically significant cutoff for the IHC score. Further, when you look at the IHC 0 cases, whether by mRNA expression or by more sensitive proteomics methods, there's a subset of cases which still express some amount of HER2. And it's possible within these IHC0 cases, there's enough HER2 expression in some of these patients that they may still benefit from some of these newer antibody drug conjugate therapies. So very briefly, what we did is identify an H&E based signal within IHC0 categories that would identify cases that we have classified as being as HER2 negative as they possibly can get. They were dead negative by IHC, so IHC0 cases, and they were dead negative by mRNA expression as well. And that's what we call true HER2 negative, because that's a case that's unlikely to have enough HER2 expression to even be identifiable by these new ADCs. And so when we did this, what we found is that we could identify those cases within IHC0 with a very high degree of confidence. The AUC here, again, is about 0.91. And so that leaves us with a very interesting hypothesis here, because in these IHC0 category, we now think that the cases that on the H&E have been identified as likely HER2 negative, by definition, are going to be enriched for true non-responders to some of these therapies, while those that still have residual expression, and there is a population within IHC0 that this algorithm identifies, that look more like cases that should have stayed HER2 low, those might be enriched for responders to these therapies. And that's something that we are actively pursuing in terms of clinical trial testing, to see if that hypothesis continues to hold up, and this can be used to guide therapy for some of these new agents. So to just briefly wrap up, our overall approach to computational pathology involves looking for diagnostic, prognostic, and predictive algorithm development, which is based on massive data sets that we have available to us, as well as novel machine learning approaches, where we're looking for prognostic, predictive, as well as diagnostic biomarkers. And we have a digital pathology platform for algorithm deployment to try and ease adoption of this that is scatter agnostic, it's AI enabled, and can disseminate both Paige-developed as well as other pathology AI. And ultimately, what we hope to do is empower pathologists to transform the way they extract clinical information from tissue samples, transform diagnostics, and ultimately improve patient care. With that, I think we can turn it over to our colleagues at PathAI.
Yeah, thank you so much, Joe. I wanted to bring everyone back on screen and just digest a lot of really extraordinary case study information which you shared. Thank you so much for that, especially for sharing the deep science. And the question as a bit of a layperson that I want to ask is, with the richness of your findings, with the work you're doing, why, in fact, is the adoption of digital pathology and some of the efforts you're making, why is it so slow? You would think that pathologists would be running towards this. Especially, I wanted to open that question because I think just about all of you on this call are pathologists. What's the barrier? I'll open that to anybody who would like.
I can speak to that, although Mike, I don't know if you want to jump in here.
Yeah, well, I mean, that's the $1 million question. I mean, we've been asking that question for decades. The digital pathology is not new. The AI part of it is somewhat new. And image analysis even is not quite that new. So I have, my theory is the reason it hasn't been adopted widely is that the value has to be there. Clinical value all medical technology has to demonstrate a certain level of clinical value that alters patient outcomes at the end of the day. Otherwise, you're dealing with, I think, just straight efficiency plays and other sort of forms of value that do exist and are helpful in the healthcare space. But those types of value have much slower adoptions than the type of value that is created when patient outcomes are genuinely altered for the better. And so what I think the work that Paige just demonstrated and some of the work I'll talk about really is trying to get at, is our technology going to be able to help patients? And is that clear and robust in such a way that I think we'll see, better adoption in very, soon, if not. And David, Dr. Klimstra, I'm sure can talk to this. You guys are seeing some adoption, I'm sure already as we are as well.
Yeah, we are. I mean, it is economics, Anne-Marie. I mean, unfortunately, I hate to say that, but setting up a digital scanning operation is very expensive. Scanners can cost up to $250,000 a piece, and you need multiple of them to digitize a pathology department. You have to get everyone to change their workflow. And so there has to be some value beyond, as Mike said, there are certain efficiencies afforded by the digital environment, but it's really hard to justify that transformation without the kinds of tools that we're discussing. And so we hope as they become better recognized and used and asked for by biopharma, by patients, by clinicians, that this will lead to reimbursement for them, which will lead people to adopt the technology to derive those data.
It sounds complex, and yet in a world of precision medicine, where N of 1 is fundamentally the baseline here, this seems to be spot on in terms of the kind of technology that we should be using, with some of the early case studies proving that cost savings, patient benefit, better targeting, et cetera, et cetera. So all signs are a go.
If I can add as well to what Mike and David have talked about, to change the course of medicine is not, a single study in these indications. It's a volume of data, right? So we in medicine, I think, are reluctant to change our clinical practice patterns without that plethora of data that support adoption and utilization efficacy of these devices. As this evidence builds, we'll be able to incorporate those into clinical practice guidelines and recommendations from the societies, which will further enhance that adoption. But, as the course of medicine is a bit of a super tanker and we're firing spitballs at it with each of these pieces of evidence, it's going to take some bigger data to move the course of that super tanker.
Thank you for that analogy. And David, I want to throw it back to you with one final comment before we move on to our friends at Path, is that just a little bit more about I believe I heard the Paige prostate just received a FDA approval, which in fact generalized the approval of the use of AI and pathology in general. Could you just clarify that for me? It sounded like
not exactly.
will actually approve as part of this.
So they have authorized the use of this algorithm as decision support for the reporting of prostate biopsies in routine practice. In the process of doing that, they essentially have defined our device in a very broad way, suggesting that the parameters that were used to approve this device would be employed for any other similar AI diagnostic algorithms that come before their review. So it provides a framework for others to follow. If anybody wants to know how to get an AI approved by FDA, there's a decision summary you can read and understand exactly what was required. And so, I think that it was an advance to get an approval, but it was also nice to know, okay, here's how this is going to work going forward.
Thank you for clarifying that. And it was a wonderful build off of what Cristin said, is, pushing the penny forward constantly. Just this one singular approval, I think, has opened a framework and a door for others to follow. So I'm going to pass it over to Mike and to Cristin for their presentation, then we'll all come back again together.
Great. Okay. Well, again, thanks, Emery, for inviting us at PathAI, and thanks, David and Joe, for your setup to my talk. I think I'll be underscoring a lot of the consistent themes. So I'll go through a little bit of who PathAI is for the audience, and a couple of case studies to try to illustrate, I think, the value and the power of the technology, and particularly in relation to how it could improve patient outcomes, and a little bit about making it real. How do we take this forward into clinical practice and make it sort of available? So very briefly, PathAI, we were founded in 2016. We're headquartered in Boston. We have about 800 or so employees. A large group of those employees came through an acquisition of a very large anatomic pathology lab that's headquartered in Memphis now. We're venture backed and backed by several strategic partners, including several pharma partners, as I know Paige is as well, which just speaks to the power that both pharma and the general clinical community see in this technology. Our vision is to improve patient outcomes. This really is at the heart of what we do. We do this by unlocking the potential of biomarkers, as we heard a little bit about, commercializing that science at scale, and ultimately improving the accuracy and the time to treatment for patients, which is really what we want to do. And why is that important, and why haven't we been able to do that? I think this is an interesting graphic on the left, which shows the drug approval rates in relation to the amount of dollars spent. And even though we're spending a lot more dollars and we're getting more drug approved, we really, on a per dollar basis, are not getting more drugs approved in an efficient way. That study, which was published in Nature Reviews, went to 2010, and then next to that, I'm showing sort of an update of that in 2018, and we still, even in the past couple of decades, have not really improved that. AstraZeneca did an interesting look back at their pipeline, which I'm showing on the right here, where they looked at reasons as to why they had failures or failure to progress, or studies that were closed in the clinical development space, and both understanding mechanism of action in humans. So it's a lot of mechanism of action work that happens in animals and preclinical work. When you translate that into humans, it's where it's very difficult to do and to learn what is happening within the human. So understanding the mechanisms of improving it, as well as selecting the right patients, both of those were huge factors. And you can see 74% of their drug pipeline, when they didn't understand mechanism of action in humans, closed clinical phase, and 56% of those programs also closed if they didn't have the right selection strategy of patients. So we're still need to do much better in terms of technologies that we can apply into that late stage clinical space. We believe that pathology has a very important role to play. There's been a lot of work in the molecular space, next-gen sequencing space, a lot of work in imaging and anatomic space, and pathology has lagged in their ability to add important amounts of information into this process for reasons that Dr. Klimstra talked about. You know, it's an old technology, it's laced with inter-reader variability, and it's not scalable. And for that reason, it's really lagged in terms of the amount of information that we can provide into these drug development programs. And yet, disease is defined by pathology. It is disease in many ways. It's been written in hundreds of years of hematoxin and eosin, as we like to say. So understanding pathology is important, capturing the disease manifestations, which underpins these molecular alterations, and really understanding what's happening at the level of the pathophysiology is something that we need to do a better job. And of course, AI, we believe, can really help in that way. Like our colleagues at Paige, we sort of see this in two ways that artificial intelligence can drive our advances in pathology. One is to enhance what we are already doing, so making it much better. These are just showing heat maps of a computer that can automatically detect the tumor region and the stromal region within a slide. And we can reduce the intra and inter-reader variability. We can improve the accuracy of patient selection, as we saw, and standardize the assessment of existing biomarkers. I'll give an example of that. But also what we can't see. As Dr. Oakley said, there's huge amounts of information in what looks like a very simple tissue section, and we can pull and extract that information out using artificial intelligence and identify new biomarkers of response, new understandings of resistance and mechanisms, and we can apply even more sophisticated unsupervised types of methods like graph neural nets. We can show us the spatial locations of these cells, which gives us a lot of functional information that we otherwise weren't able to get in the past. And we can use that to find new mechanisms and potentially new drugs and new selection strategies. So I'll talk a little bit about kind of both of those areas. This is, again, just how we're set up as a company. We lead with the science. Important to pull that through into clinical trials, into a scalable way that we can kind of do this globally, and also then bring it into places for patients, of course. So I'll sort of skip over that quickly and just talk about some of the exciting science. I'm going to give a couple of examples here. One is in the space of immuno-oncology, PD-L1 or anti-PD-1 therapies, for which Jim Allison won a Nobel Prize not too long ago for our work in IO. We know that this has been very successful in many different cancer types. And patients who are eligible, in some cases, for anti-PD-1 therapy are tested for PD-L1 expression and a bit of a continuous scale. So if you have a high PD-L1 status, you're very likely to respond to this drug. And if you have a low PD-L1 status, you're unlikely to respond to the drug. And pathologists make that measurement under a microscope, and it's highly variable, as I'm showing here. This is showing the agreements of pathologists in reading PD-L1 at different levels. So at 1%, how much they agree, at 50%, how much they agree, and also looking at PD-L1 in immune cells where they disagree very poorly. And so in the tumor space, it's borderlines on weak agreement, and certainly when you're measuring in immune cells, it's very weak agreement. And so what does this mean? It means that patients are getting misclassified in terms of whether or not they can be eligible for a therapy, which is unfortunate in the case where patients can be denied therapy, or in cases where patients get therapy where we know that they would not respond and are subjecting them to potential safety risks as a result of that. So we developed AI models that can measure PD-L1 across the whole slide, every single cell. We can determine whether or not it's PD-L1 positive or negative, what cell type is positive or negative, and where it is within the cell, within the tissue, I mean. And so we developed AI-based models to do that automatically, and then we wanted to apply that at scale across many different retrospective cohorts to see if it actually mattered and impacted the outcomes in these studies. And here I'm showing one. We published this in Modern Pathology recently. I'm showing one interesting observation by using AI instead of manual reading. One is that you can increase the biomarker prevalence or the biomarker positivity rate in patients, particularly at low cut points. So we found that there were about 20 to 30% more positive patients when we used AI, which means it's much more sensitive at detecting the biomarker. We saw this with what Joe was talking about with HER2 low. It's a much more powerful technology at these very low thresholds. But does it matter? And are those patients actually responders to the drug? So when we looked retrospectively at patients who were treated with Opdivo or nivolumab, this is a Kaplan-Meier curve for those uninitiated, which is basically showing the survival over time of patient groups. And patients that are negative for PD-L1, both manually reading as well as the AI, so consider them true negatives, do the worst. Patients who are doubly positive in the blue line do the best. But those patients, those 20 to 30% of patients who were negative by manual reading, who in the real world would have been denied therapy, turns out that they have a very strong response rate similar to patients who are double positive. So these are, in fact, patients who could respond. And when we looked across many trials, we found that 10 out of the 13 trials we looked at across several different tumor types, melanoma, bladder, lung, that we were able to increase the significance of the drug effect in the patient populations that had the AI reading. So again, we're showing that we can find more patients and that those patients potentially are responders. And so we think that we have a better tool here to give patients the best course of treatments. So that's one example. Another example in this space of what about things that are new? So PD-L1 is an existing biomarker. Here we're looking at CD8. CD8 is a cell type that is known to be involved in clearing tumors or attacking tumors. It is what we're trying to turn on in immunotherapy to get our bodies to attack tumors themselves. And we have known in the literature that patients that have inflamed tumors, a lot of immune cells there, they do well in immunotherapy. But there are some patients who are desert, have no or low immune cell infiltrates, and some who have immune infiltrates, but they surround the tumor. For some reason, they are not able to get into the tumor. And this has been very difficult to study across cohorts because it's hard to measure CD8 under a microscope. You get a lot of interreader variability, and it's hard to scale this and standardize it. So we developed a model to measure CD8 and to classify into these desert or excluded or inflamed phenotypes, and then apply that classification of each patient and retrospectively look as to whether or not they were responding to immunotherapy. And so when we went and we looked at a study, again, we did in collaboration with Bristol Myers Squibb, you remember, again, if you have PD-L1 status high, you respond well, and if you're low, you don't respond well. So we already knew that. This was data from their existing clinical trial. But when we looked at CD8 excluded patients in the PD-L1 negative population, that's this line down here, these patients are poor responders, we found a subpopulation that responds very well, both to monotherapy and to combo therapy. And when we added that group of patients to the PD-L1 positive group, we saw an increase in the biomarker positive patient population. So again, potentially to add more patients to get life-saving therapy. And the hazard ratio also gets better, meaning we have a more powerful drug effect from an efficacy perspective. So new biomarker, adding it to existing biomarker could really improve patient outcomes in this case. So those are the IHC examples. Joe actually gave a really good examples of how you can predict molecular phenotypes from H&E. We've done very similar work that we published in Nature Communications, where we, instead of doing end-to-end, where we just feed in a slide, we have, I didn't mention this, we have about 450 pathologists who work with us part-time to help us develop our models. And they can sort of annotate these different tissue regions, and we can use that in a structured data way to get all this information out of H&E, and we can use that information to, for example, separate in a clustered way gastric tumors from lung tumors, for example, from skin tumors. We can look at these features of histological features and associate them with various tumor types. For example, we can see that macrophages in the cancer-associated stroma are highly associated in stomach cancer, in lung cancer, as one might expect. And we can use that information then to further predict molecular phenotypes, just like we saw Paige was talking about in the EGFR and MSI. Here we're showing that we can predict with a reasonable area under the curve HRD, which is another molecular drug target, and we can predict that from H&E. And so why is that? Actually, we can go even further, and we can kind of link pathology phenotypes to molecular phenotypes, which gives us a much richer understanding of disease mechanisms, linking these two things together, which is some work that we've done recently and sort of dug a little bit deeper into why our HRD predictions are working. But why does it matter? And I think that Paige touched on this as well. H&E staining is cheaper and more accessible. Everybody gets it. Next-gen sequencing is expensive. Not everybody gets it. The turnaround time is long, and the failure rate of those tests are high. So if one could predict these molecular phenotypes from H&E, we can increase access, we can decrease costs, we can decrease patient leakage for tests that are on the market, and we can also help pharma companies find more patients to enroll into their trials for the same reason as we heard about earlier. Patients get diagnosed with cancer, and in today's world, they go directly to biomarker testing. A percentage of them go to biomarker testing for the reasons that I said. But if you can use an H&E very quickly and very cheaply to triage these patients and enrich, as we saw Joe was talking about, we have a higher likelihood of bringing those patients into our trials and potentially enriching in that way. So certainly a very exciting development in that space. So then the last thing I'll talk about very briefly is how do we bring this forward? It is important, I think, for algorithms that we're developing to live seamlessly from research to clinical trials to diagnostics. The clinical trial space has their own unique needs. When we're deploying an algorithm in a clinical trial, we might want to look at study-specific protocols, very trial-specific things like I want 3 pathologists and not 1, or I want a central reader, or I want overreads, or I don't. The sponsor wants to see it in real time. There's a whole host of things that clinical trials are demanding, whereas the clinical space is less demanding on that, but more demanding on needing to see patients longitudinally over time, and there's no real trial-specific things there. So we've developed these platforms that go from research to clinical trials to diagnostics, and in doing that, we're able to deploy these platforms either in our own lab services, where we can get slides sent to us. And Anne-Marie, you were touching earlier that why hasn't adoption happened? And the fact is it hasn't yet. So there's a lot of labs out there that don't have imagers. And if they don't have imagers, they can simply send glass slides to a lab like ours or to another central lab. Slides are sent, I'm sure, David, as you know, all the time, every day, a huge portion of them. And so shipping glasses isn't terribly difficult. But also, we can have our platforms at CRO sites or at clinical sites as well for those labs that have digitized. So we're trying to meet, I think, the community where they are in terms of providing as best access as we can to the technology going forward. And I think I'll just end it there. Sorry that there's only maybe 11 left, but hopefully you get a sense of where we're headed.
Please don't apologize. It was an extraordinary presentation, actually, from both sides, and I'm realizing now that an hour isn't even touching the surface on such an important conversation. So don't worry, we're going to invite you back for our next conference. I want to before there's a whole range of questions here coming in, but before we do so, I just want to get into a very specific conversation on, we understand the barriers here, but I could, from both of your perspectives, could each of you summarize, the top 3 to 5 benefits of AI and how it's fundamentally shaping the future. I realize that's a big.
Oh, you're on mute.
And David, yes, there we go. David, I'm sorry we are on mute. Yeah.
That's my fault.
No
Sorry.
worries.
I think the talking in regard to pathology in specific, the benefit of AI is that there's a potential for computer vision to see things that pathologists can't. As Joe Oakley mentioned, I mean, the tumor morphology is an integration of all sorts of things that are happening at the genomic level, and so it's capable of assessing that and synthesizing it and coming up with endpoints that we can't derive as practicing pathologists. It's also capable of teaching us what those are. So you can introspect the AI and ask the computer to show you what is the basis to distinguish HER2 expressed from HER2 not expressed. And you can begin to understand what these morphologic features are. And third thing is you can apply it to a routine tissue to derive information that would typically require, thousands of dollars of testing, a quantity of tissue that may not be available and take 2-3 weeks to generate results. So those are my three.
Mm-hmm.
Yeah.
Mike, do you want to add?
Sure. Yeah, of course. Yeah. I mean, I think, scalability is a big one. So, pathologists do what they do very well. I think we shouldn't look at AI and saying, hey, we're going to get rid of pathologists. I think 95% of the time they do what they do very well and they don't really need any help. It's when we're asking pathologists to quantify things where it starts to fall down, that subjectivity comes into play. How gray is gray? And that begins to matter if you're selecting a therapy. And so the computer can just do a much better job. And so number one, it can be standardized. Emery, you go to a certain pathologist, whereas I may go to another, and we may get different results because we've gone to two different readers, or we went to the same pathologist, but one read it in the morning and one read it late at night. And so we get different results. The computer won't do that. It will be standardized and it will be the same every time. That's very important. And I think in these quantitative regions that are very difficult for a human to do well, I think we can see computers doing much better. So that's some of the results that I showed. Just the accuracy, I think, is a very important part of it. Again, for these limited cases that are very important cases, but limited use cases where I do think that it would be very valuable. So I'll stop there. Maybe Cristin, you have some of your own advantages you think for AI.
Yeah, I think, Mike, I agree with you on accuracy and value in precision medicine. I think that's the number one thing that comes to my mind. In addition to that, I think that AI can really be utilized as a tool for equity and for access in medicine, and showing the value in those populations that have restricted access to good pathologists, to subspecialized pathologists. This might be a tool to leverage the equity of care in those populations as well.
Yeah. Thank you for reinforcing that point. I'm going to go to a number of audience questions that have come in. So let me read them to you. And the first question is, it took so long for genomic comprehensive profiling, or what's known as NGS, to be implemented across institutions. Is AI a replacement for that, or does it complement it? And I'm happy to just have someone put their hand up and take the question.
Joe.
Go for it, Joe.
I'm happy to take that question because my background, my fellowship was in molecular genetic pathology. So bringing up a lot of that genomic analysis, it was a big part of my fellowship in my early clinical practice, and then as well as what I was doing in industry. And so the question is often, it's when we get a lot, and it's sometimes framed in a different way. With next generation sequencing doing everything, what is for AI to do? And what I would argue is that, as I said, it's an ability to do phenotypic analysis. It's an ability to quantitate, as our PathAI colleagues have filled in. And so what you're getting really is a functional complement to the genomic information that you are getting. And I think, a really great example of this is something like FGFR mutations within bladder cancer, with FGFR3 has a really bad habit, because it's a very promiscuous translocation partner, of winding up as an N of 1 or N of 2 on all of these bladder cancer studies. And so it's not clear among all of the many translocations that have been seen how many of them are actually functional. But you might be able to identify a signal that is consistent with activation of FGFR. And so you can marry that to the genomic information to not only identify those cases that have the genomic alteration that you're targeting, but are actually using it in a way that the drug is going to address. And remember, the drug doesn't treat the genome, it treats the phenotype, the expression of that genome. And that's where I think the two techniques combine.
Right, but so very complementary in general. So a build on that question is, with the advancement of AI in pathology, does that implicate finding more biomarkers in cancer and therefore developing more targeted therapies?
My short answer is yes. I just
Yeah.
think by virtue, it's a whole new data source, and it's a robust data source. It's new, and there's so, as I think both of our talks kind of show, there's so much more information that we can glean. So it certainly should open up new mechanisms, new understandings, and potentially new targets, new strategies, new indication strategies, even. Help us really understand which patient population to go in up front.
Yeah, it seems to be the ultimate goal of both the work you're doing, really.
Yeah, I think I would amplify that further to say that it allows us to look at existing genomic biomarkers in a different way. Building on what Joe said, I mean, we're often drugs are targeting the activation of an oncogenic pathway, which may involve 2, 3, 5, or many genes and other alterations along the way. But if they all produce a similar morphology, we may be able to find that as a single common biomarker, digital biomarker, that reflects the cancer pathway that you're trying to target. And so in some ways, it could actually simplify biomarker detection by bringing it back to the morphology that is a reflection of that pathway abnormality.
Mm-hmm, mm-hmm. So we only have a couple of moments left, but I wanted to finish on a final question, because I know that our audience very much appreciated the case studies that both of you presented, and it struck me that a particular one starts to talk about biomarkers. We're looking at prediction, we're looking at diagnosis, we're looking at treatment applications. So really across that entire spectrum. Where do we think the future of AI and biomarkers actually is? Is it more balanced on the treatment side? Are we going to see things develop more on treatment? Or are we going to see more develop on predictive? Is there any kind of understanding of what's going to happen first and what's closest in here?
Maybe I'll go first on that. I'm not sure I separate those two things. I think if we're talking about diagnosis, as I think what David opened up with, for example, better prostate and better breast, for example, diagnoses, those certainly are nearer term in a way that they're immediate applications, something that can be implemented literally today. Some people are implementing today. On the other hand, I think that these precision medicine approaches that relate to very highly impactful changes in treatment paradigms, while a little bit further away, I think are going to gain an enormous amount of investment and traction in the not so distant future because of the kinds of impact that they can have. So that's a little bit of a non-answer for you, but I think that's how I might look at it.
Yeah. It was a hard question, actually.
I think it's spot on. I mean, at the end of the day, the whole purpose of pathology analysis is to guide therapy. I mean, yes, you want to know what the diagnosis is, you want to know what the prognosis is, but at the end, what you're really trying to determine is what is the next step in the treatment of this patient. Do they need surgery? Do they need chemotherapy or not? If they need chemotherapy, what drug do they need? And so all the information that we can bring to bear about the prognosis or the response to a drug is heading us in that general direction of predicting the best treatment for this patient. And you're talking about N of 1. I mean, that's every cancer is unique. And so we need to be able to do this very broadly.
Mm-hmm.
The
Thank
other
you,
thing
David.
I would-
Go ahead, Cristin. I'll give you the final word.
The other thing I would just add to that, I think that the approach that PageAI has taken, as well as PathAI, is really about looking at those unmet needs today, right? And having those unmet needs guide us into where our areas of development are. So if there is a value statement in a particular disease for treatment or for prognosis, I mean, that is where we are looking to add value to patients.
Thank you.
Yep.
I'm going to leave it there, and with many thanks on behalf of our audience and Colorectal Cancer Canada, we appreciate you being here. I realized that I think we just started to scratch the surface, and this in itself could be an entire conference. So thank you all for being here. I encourage our audience to investigate both of these companies. Both of them are fast tracking some magnificent technologies. They're really exciting companies. So please do check them out behind the scenes. And thank you again to our team.
Thank you. Thanks so much.